Welcome back. Physics builds models. Chemistry builds structures. Biology builds evolutions. 3 ladders, 1 way of thinking. And today, we drop that thinking straight onto a real exam idea. Today's focus: DNA manipulation and applications, STRs, gel electrophoresis, restriction enzymes, plasmids, GMOs, viral vectors. Scientists analyzed the DNA samples of 3 students, Rosa, Li, and Abdul. Short tandem repeats, STRs, that are unique to each individual were investigated. Each student's DNA was digested using a restriction enzyme. The length of each STR for each student is shown in the diagram below. We are given the STR fragment lengths for 3 students, Rosa, Li, and Abdul, and we need to determine which lane on the gel belongs to whom. The core logic here is incredibly simple. Smaller DNA fragments move faster and travel further through the gel. By sorting the fragments from largest to smallest, we can map them to the wells instantly. Abdul has both the largest, 1000 inch BP, and the smallest, 30 BP fragments. This means his lane must feature the highest band and the lowest band on the entire gel, which perfectly matches well 3. Li has 2 relatively large, intermediate fragments, 800 inch BP and 500 BP, which corresponds to the higher, closely spaced bands in well 2. Rosa has smaller fragments, 400 inch BP and 60 BP, meaning her bands will travel further down the gel, matching well 1. Therefore, reading from wells 1, 2, and 3 respectively, the correct sequence is Rosa, Li, and Abdul. The correct answer is D. We have a circular plasmid with a total length of 80 inch kbp, and it is treated with 2 restriction enzymes, EcoI and BAMHI. Our goal is to determine the total number of fragments produced and their exact lengths. The biggest trap in this question is the presence of hind III. Since it isn't added to the reaction, we must completely ignore its restriction sites at 20 inch kbp and 65 kbp. Let's locate our active cut sites going clockwise around the circle. EcoI cuts at 80 inch kbp, which we can treat as 0 kbp and 70 kbp. BAMHI cuts at 15 inch kbp and 40 kbp. Because a circular plasmid is cut at 4 distinct positions 0, 15, 40, and 70 inch kbp, it will yield exactly 4 fragments. Now, let's calculate the length of each fragment by finding the distance between consecutive cuts. From 0 kbp to 15 kbp equals 15 kbp. From 15 kbp to 40 kbp equals 25 kbp. From 40 kbp to 70 kbp equals 30 kbp. From 70 kbp back to 80 kbp equals 10 kbp. Arranging these from smallest to largest gives us fragment lengths of 10, 15, 25, and 30 kbp. The correct answer is B. Let's move on to the next question. This question asks us to identify the defining feature shared by all genetically modified organisms. The key to solving this instantly is understanding the distinction between a GMO and a TGO. GMO, genetically modified organism, any organism whose genome has been modified using genetic engineering technology, e.g., editing, deleting, or altering existing DNA. TGO, transgenic organism, a specific subset of GMOs that contain DNA foreign to their species, i.e., foreign DNA introduced from another species. Now let's evaluate the options using this distinction. A and C describe transgenic organisms, TGOs, where foreign genes or non-native proteins are introduced. While all TGOs are GMOs, not all GMOs are TGOs. D describes a specific gene knockout technique, which is only one of several ways an organism can be genetically modified. B captures the broad, fundamental definition of a GMO, any organism that has at least one section of its DNA altered by scientists using genetic engineering techniques. Therefore, the correct answer is B. The flow chart outlines the complete process of recombinant DNA technology, gene cloning, across 6 key steps. Step 1, isolation donor, human, DNA containing the target gene and vector plasmids are isolated. Step 2, cutting and ligation. The donor DNA and vector plasmids are cut using restriction enzymes and joined together, via DNA ligase, to form recombinant plasmids. Step 3, transformation. Host bacterial cells take up the recombinant plasmids. Step 4, bacterial proliferation. A gene cloning. The transformed bacteria undergo binary fission on nutrient agar, replicating both themselves and the recombinant plasmids inside them, making many copies of the target gene. Step 5, selection and scale-up. Successfully modified bacterial colonies are identified, isolated, and cultured in large tanks or bioreactors. Step 6, harvesting. The protein products expressed by the bacteria are harvested and purified for downstream use. Looking at question 37, we are asked to identify the correct statement regarding the 6-step procedure shown in the flow chart. Let's analyze each option step by step. A is incorrect. Donor DNA comes from a human cell, but vector plasmids are isolated from bacterial cells, not human cells. B is incorrect. In step 2, the same restriction enzyme must be used to cut both the donor DNA and the plasmid so they produce complementary sticky, or blunt, ends that can anneal. C is incorrect. Step 3 illustrates bacterial transformation, plasmid uptake, not transcription, RNA synthesis from DNA. D is correct. In step 4, as the bacterial cells divide by binary fission, the recombinant plasmids containing the donor DNA also replicate. This process creates multiple copies of the donor DNA, gene cloning. The correct answer is D. Step 6 of the process involves harvesting and purifying the protein products synthesized by the genetically transformed bacteria. Let's evaluate how these products are used in biotechnology. A is incorrect. Inserting genes into viral vectors to treat conditions like cystic fibrosis refers to gene therapy, which delivers functional genes directly to human host cells rather than harvesting proteins from bacteria. B is incorrect. growing tissue or organs from stem cells involves stem cell technology and tissue engineering, not bacterial protein synthesis. C is correct. Recombinant DNA technology in bacteria is widely used as a molecular factory to produce commercial quantities of therapeutic human proteins, such as insulin, human growth hormone, HGH, or blood clotting factors. D is incorrect. Breeding transgenic animal models involves mammalian genetic engineering and selective breeding, not harvesting bacterial proteins. Therefore, the correct answer is C. Citrus greening is a disease that affects citrus trees, such as orange trees. The disease is caused by the bacterium Candidatus Liberibacter asiaticus. These bacteria are transferred to the trees when insects called psyllids feed on the sap in leaves. The bacteria live in the plant's nutrient-conducting tissues, phloem, causing slow death of the trees. A solution to this disease uses a gene from a spinach plant, which codes for a defensins protein. The defensins protein binds to and punches holes in the bacteria, breaking them apart. Genetic engineers have inserted the defensins gene into a viral vector. The viral vector is a modified form of a virus that normally infects citrus trees. The genetically engineered viral vectors were placed in many orange trees through small incisions in the tree's bark. After several years' growth, all of the treated trees were very healthy, while all of the untreated trees nearby were affected by citrus greening. Disease context. Citrus greening is a fatal plant disease caused by the phloem-dwelling bacterium Candidatus Liberibacter asiaticus, transmitted by insect vectors called psyllids. Mechanism of action. A gene encoding a defensins protein isolated from spinach is used. Defensins destroy the bacteria by cell lysis, punching holes in them. Delivery system. Genetic engineers engineered a modified citrus virus to act as a viral vector, carrying the defensins gene into orange tree cells through physical incisions in the bark. Outcome. Expression of the gene enabled treated trees to produce defensins and remain healthy. Question 39 asks us to identify the specific role of the viral vector in this genetic engineering solution. Recall that the fundamental purpose of a vector, whether a plasmid or a virus, is to act as a delivery vehicle, transporting a foreign gene of interest into host cells so it can be expressed. Let's evaluate the options. A is incorrect. The viral vector itself does not kill the bacteria. It is the defensins protein expressed by the host plant that destroys the bacterial cell membranes. B is incorrect. The system targets the pathogenic bacteria in the phloem, not the psyllid insect vectors directly. C is correct. The viral vector is a vehicle designed to deliver viral nucleic acid carrying the spinach defensins gene into the citrus tree cells, enabling the tree to produce its own protective defensins protein. D is incorrect. The viral vector is modified in a laboratory setting to carry the gene, not by invading spinach leaves in the wild. Therefore, the correct answer is C. This question looks at the broader agricultural and environmental advantages of using a genetic engineering solution for disease management. Without genetic engineering, farmers typically rely heavily on spraying massive amounts of insecticides to suppress the psyllid insect vector that transmits the disease. Let's evaluate the options to find the true advantage of this genetic approach. A is incorrect. The defensin's protein targets bacterial membranes to protect the tree. Its purpose is disease resistance, not enhancing the nutritional profile of the fruit. B is correct. By equipping the trees with internal defense mechanisms against the underlying bacterial infection, farmers no longer need to rely on increased or heavy insecticide applications to control psyllid populations. C is incorrect. Single antimicrobial proteins like defensins do not alter the taste profile of the fruit or produce spinach-flavored juice. D is incorrect. Genetic engineering techniques frequently raise public or ethical concerns regarding GM organisms. It certainly does not avoid public scrutiny. Therefore, the correct answer is B. STR analysis and gel electrophoresis. Individuals show unique STR lengths. Restriction enzymes cut DNA at specific sites. Fragments separate by size in gels. Example from document: STRs were marked with a fluorescent probe and were the only visible bands in the gel. Restriction enzymes and plasmid mapping. Cutting plasmids at multiple recognition sites produces predictable fragment numbers and lengths. GMO versus TGO definitions. GMO, any organism with DNA altered by scientists. Quoted, have at least one section of their DNA that has been altered by scientists. TGO, contains DNA from another species. Recombinant DNA in bacteria, steps. Isolate DNA cut with restriction enzymes ligate into plasmid transform bacteria select colonies mass-produce protein. Quoted, Step 6 bacterial products are harvested and purified. Application, production of human insulin, HGH. Gene therapy and viral vectors in plants. Spinach defensin gene inserted into citrus infecting virus. Virus delivers gene into tree cells. Tree produces antimicrobial protein protects against citrus greening. Quoted, deliver viral DNA with the defensins gene into the cells of the orange trees. Key advantages highlighted. Genetic engineering can reduce insecticide use by giving plants internal disease resistance. At the end of every lesson, I want to leave you with one idea: learning becomes powerful when you climb the logic ladder. Physics moves from situations to models to equations. Chemistry builds from structure to property to reaction. Biology grows from variation to adaptation to survival. Different subjects, but the same underlying logic. When you learn to decode systems, build ladders, and construct logic, you don't just solve problems, you understand how they fit together. If you'd like to keep climbing this ladder with me, you can find more lessons, breakdowns, and reconstructions on my channel. Thanks for learning with me today.